Hi, welcome to the lecture on environmental adaptation of animals, and now we will look on the animals in different environments. First on the aquatic and then on the terrestrial animals environments. And in the aquatic environments, we always have plenty of water around us, but the difference is that in the ocean and in the lakes, there might be different salt concentration, and therefore it might be sometimes difficult to get the water to your body. Let's look on the sea, where the osmotic pressure of these marine fish is pretty much the same as in the humans. So maybe 300 milliosm per, but there also might be some that are having a little bit higher. But anyway, there's two times more solutes in the ocean. So the osmotic pressure is about 1000. So therefore the environment is hyperosmotic. And in the hyperosmotic, there's risk of two things. One is diffusion of ions, and what the other is osmosis of the water. And these are quite fundamental challenges. So if the animal is fighting against diffusion and osmosis, it must use energy. And let's look on the freshwater animals, where there is also water available, but the problem might be a little bit different. So let's look first. We have different kind of animals, and they have different kind of osmotic pressure. So freshwater mussels and snails have very low osmotic pressure. Then the crayfish has a high, and then mosquito larvae, brown trout, frog, etc. They have pretty normal, so 300, 200 milliosm per kilogram. But in any cases, the environment is hyperosmotic. So it means that they have much more solute inside the body, inside their cells, than in the environment. What kind of ions they have? Well, they have all ions, not only one or two, but all ions are much more in their blood or in their body fluids than in the river water. And its difference might be that it's only twofold, or it might be that it's 400-fold. And what is the difference then? Okay, freshwater mussel and snail, they have low sodium, they have low chloride. And then this crayfish is having very high sodium and chloride. Otherwise, there is not huge differences in the ion concentrations in other ions. But anyway, they all have, in all these ions, they might have 2 times more in the body fluids or 400 times more in the body fluids. And therefore, they are all the time working actively to prevent the accumulation of water and to prevent the loss of ions. So how they can prevent the water accumulation and losing the water ions is that, okay, in the blood, in this crayfish, maybe 400 milliosm, 200 millimole per liter of sodium, so much higher than in the environment. So diffusion and osmosis says that, okay, salt it's losing salt and it's gaining water. And we can simply understand that, okay, this flow depends on the osmotic difference, so the osmotic gradient and the ionic gradient. So for example, in the sodium, there is 400 times more sodium inside this animal, so it's definitely going to be outside if there is a road. if there is a sodium channel, if there is any kind of transporter that can mediate sodium outflux, it will definitely go. And from that, we can get on this second term of the flow rate, so the permeability. So if there is no sodium road, then you can say keep the sodium inside your cells. So the permeability of the animal surface is very important. And of course, then the surface area of the exchange. So if you have different kind of areas where somewhere you are allowing water and sodium to flux through the cells epithelium, then the surface area is important. But let's see, in the theory, in the diffusion and osmosis, that this is the risk, but you can get rid of it a little bit with active transportation. So you can get sodium and chloride with the gills. You can get also salt in the food, and then if you are regulating how much salt you are releasing in the feces, then you might prevent this losing the salt. But also your body might be different from the ocean crayfish. So these freshwater animals usually have a little bit less concentrated bloody fluids. So then the osmotic and ionic gradient is smaller. And the integument, so the animal surface in the freshwater animal, is usually less permeable. So if you are minimizing this osmosis and the diffusion with the exoskeleton or good skin, etc., then you are minimizing the flow. But the problem is that when you are breathing, you anyway, there's a risk of losing water or getting too much water, etc. Okay, and we also have nitrogen waste, and we got to get rid of it. So in these freshwater animals, typically they are peeing all the time. Cloud toad is producing 60 milliliters per 100 gram of the animal urine. So it's peeing half of its body mass every day. And of course, it's diluted drastically to maximize the water excretion. So there's the osmotic pressure is maybe 10% of the plasma, and it's also the sodium concentration is maybe 10%. So 90% of the sodium is taken back, and therefore it has more water. But the problem is that if you are peeing half of your body mass, then anyway there's a lot of sodium, a lot of iron loses in these aquatic animals, and therefore they must have some mechanism to get as much ions from the environment as possible. So they can maintain very high sodium concentration, very high chloride concentration in hypoosmotic environment, and ions are uptaken across the gill epithelium. There's active transport of pump. There's a pumping sodium and chloride. It's actually indirect, but anyway, pump means that okay, there's ATP, So the energy is producing ADP plus a phosphate plus some energy. So the interesting thing is that, okay, there is a two mechanism. There is a sodium transport, there is a chloride transport, and they are working side by side, but they are working independently. So there is separate mechanism. There is not a single molecule that is moving sodium and chloride together, because that would be a little bit difficult. And this chloride is exchanged to carbonate, actually bicarbonate. And the sodium is exchanged to proton. And how you can get bicarbonate and proton, you can use the same way as in the respiration, so the carbonic acid. And when this carbonic acid is dissociated, so one part is changed to chloride, another part is changed to sodium, and then you can get both chloride and sodium inside your cell or inside your body. And how you can get this bicarbonate is, the good thing is that, okay, this process is metabolic regulated. So it's regulated from the carbon dioxide. So more you move, more you have energy needs, then you are producing more carbon dioxide, and that will increase the uptake of sodium and chloride. How you can do it? In the gill structures, etc., we've been discussing elsewhere during the respiration, but there's the secondary lamels, and they are critical for this respiration. They are increasing the area very drastically, so the oxygen can be very effectively taken from the surrounding. But they also contain chloride cells and pavement cells that are needed for these ion uptake. And there are two proteins. There is NKCC1 that is taking sodium, potassium, and chloride, moving them together, and then, surprise, surprise, sodium, potassium, ATPase. That is the pumping mechanism. How they are working together? Well, we have the NKCC1 on this cell membrane. And through that we can get potassium, sodium, chloride together across one cell membrane. But then we must have sodium potassium pump to move some of these ions back. So there must be a third circuit, and this is the same mechanism that happens quite often in kidneys and in the digestion, etc. And so it's the transportation of sodium or chloride to get inside the cell is not might not be the critical thing of, okay, you are having it's not completely directly on the active, but you are using this sodium potassium ATPase that is using plenty of ATP, and therefore you can regulate your ion concentrations. Thank you.